Hello everyone and welcome to today's keynote presentation titled Development of an Affordable COVID-19 Vaccine Based on a Newcastle Disease Virus Platform. This webinar is part of the 2021 Microbiology Virtual Week, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Adolfo Garcia-Sastre, Professor of the Department of Microbiology and Medicine at Tisch Cancer Center, Icahn School of Medicine at Mount Sinai. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Dr. Garcia-Sastre, welcome. You may now begin your presentation, sir.
Hi, hello, good morning or good afternoon, depending on where you are. I'm going to be today showing you some of the signs that led us to explore the possibility of using a vaccine platform based on a viral vector, Newcastle disease virus, as a COVID-19 vaccine, and why now do we still need more vaccines? That's something that I will cover, and the potential advantages that this vaccine platform may have over the already existing vaccines that are in use and that actually are performing quite well. And these studies have been mainly the collaboration between three labs. It's my lab, with the lab of Peter Palese and the lab of Florian Kramer. We are all at the Icahn School of Medicine at Mount Sinai, affiliated to the Department of Microbiology, and we are also quite helpful for the contribution of different funding agencies, including NIAID, and as well as some philanthropy from the Bill and Melinda Gates Foundation, or for some other foundations like the JPV Foundation and the OPP Foundation. All right, so first, what I'm showing now is some of the strategies that have been used and are still some of them in development for COVID-19 vaccination. This includes the use of inactivated viruses. In fact, there are some vaccines that have been manufactured in China that are being used in different countries that use an inactivated SARS-CoV-2 in order to induce immunity against SARS-CoV-2, and attenuated vaccines, meaning you need to somehow make mutations in the SARS-CoV-2 virus that now the virus is unable to induce disease, but is still able to be immunogenic. And there is no attenuated vaccine in use right now. Some of the difficulties with attenuated vaccines is to achieve the right degree of attenuation, so to make them safe. And perhaps this have making the development of attenuated vaccines a longer development, but there are some that are in the pipeline that may still be used later on. And then there are vaccines that are not based on the virus itself, either inactivated or attenuated, but they are based on components of the virus. And all the vaccines that are being in use, they are based on one of the components of the virus, which is the spike protein. The spike protein is the viral glycoprotein that is in the surface of the virus that binds to the receptor, and antibodies against the spike protein, once that they are induced by natural infection or by vaccination with an antigen that has the spike protein, will prevent the infection by preventing binding of the spike protein to its receptor in the cell that is going to get infected. And that's the basis of the work of the vaccines that are in use, although perhaps there is also some contribution not only from antibodies but also from cellular immunity. But we know that the vaccines that are induce neutralizing antibodies directed against the spike protein, and that these antibodies are able to prevent binding of the spike protein to the receptor. It's also a good indication that the levels of antibodies that are induced by vaccination or by infection correlate with the efficacy. The higher the levels of antibodies, the higher the efficacy for prevention of infection with even with some of the new variants. The vaccines that are based on components of the virus, on the spike, could be a protein subunit vaccine that's expressing recombinant protein. This is, for example, the basis for the Novavax vaccine that has conducted already some larger-scale clinical trials, but it is pending of submitting the results to the regulatory agencies for potential approval use of the vaccine. And then there are some others that are based on viral vectors or in genetic material of the virus. The ones that have been successfully used in as viral vector vaccines, they are the ones that they use adenovirus vector. These are adenoviruses that, instead of expressing adenovirus proteins, express the spike protein of SARS-CoV-2. Expression only of the SARS-CoV-2 spike protein does not induce disease. For that, you need viral replication, but induce antibodies against the spike. And these are, for example, the vaccines of Johnson & Johnson, the single-shot vaccine that is used in this country, in the USA, or also the vaccine of AstraZeneca that is widely used in many countries, or the Sputnik vaccine, which is the Russian vaccine that is also used in several countries. In terms of genetic vaccines, the genetic vaccines could be based on DNA that encodes the spike protein that is being injected during vaccination, and this results in expression of RNA in the cells that this DNA is taking up, and the RNA gets converted into protein, and if the protein is the spike, then you get antibodies against the spike, or the RNA vaccines that bypass the use of DNA by using directly messenger RNA that needs to be coated with some lipids in order to facilitate the release of the RNA in the cytoplasm of the cell that is going to produce the antigen, which is where it needs to be translated into the spike viral protein. In terms of the, in addition to these platforms, one need also to consider the possibility of use of an adjuvant. That's important mainly for inactivated vaccines and protein subunit vaccines that require some help in order to be more immunogenic. The use of nanoparticles is what is the case of the RNA vaccines that need to be encapsulated into nanoparticles in order to deliver well the RNA inside the cell where it's going to produce the protein. And in terms of the route of administration of these vaccines, all the vaccines that have been approved are intramuscular vaccines. There are some that are in the pipeline that are intranasal. These may have the given advantage, if they work well, to induce very good local immunity in the upper respiratory tract, which is where infection starts. The vaccines that are in use right now induce extremely good levels of antibodies, systemic antibodies, that circulate in blood. These antibodies reach very well the lung, and and therefore in the lung will prevent infection, not the lung, but these antibodies that not reach so well the upper respiratory tract, and then replication may be still possible in the upper respiratory tract in vaccinated people. This will not result in severe disease. For that, the virus needs to go to the lung inside, but they will result in asymptomatic infections of mild infections that can lead to transmission, like what is happening, for example, in many of the vaccine breakthroughs that are happening, and that in vaccinated individuals, that's because they don't, these vaccines don't induce very efficacious, induce some, but not as very efficacious levels of antibodies in the upper respiratory tract. Well, with respect to our contribution here in vaccines, we started first with the spike and trying to understand what form of the spike we need to use in order to have good immunogenicity. And thus we conducted some studies on that. Some other vaccine platforms have conducted similar studies, but we started by looking to a spike protein that is identical in sequence to the spike protein of the virus, but then we tested as immunogens some versions of the spikes that they become stabilized without changing their antigenic properties. This was achieved either by eliminating the cleavage site, the CS or cleavage site of the spike protein. The spike protein needs to be cleaved in order to be functional, and this cleavage makes the protein less stable, so this is good for the virus. But for an immunogen, one prefers to have a very stable immunogen that does not fall apart. Cleavage may allow this immunogen to fall apart in some circumstances, and this will prevent induction of good antibodies. So we eliminated the cleavage site in one version. In another version, we introduced two prolines in the carboxyl terminal domain of the spike protein that are known to stabilize also the protein. And in the last version, we include both modifications at the same time, modification of the cleavage site and the two prolines that makes also the antigen more stable. We look them for expression, so this is using recombinant proteins. They express very well. They express in mammalian cells and in baculo insect cells also. They express nice levels of protein that can be seen purified in a Comasi gel with high levels, and they look quite stable. They don't have any degradation products. And they also recognize very well with antibodies like this particular antibody that are known to neutralize the spike. So that means that they are conformationally correct, and they are stable here in when respects to recombinant proteins. But we expect that the ones that contain the modification would be even more stable when injected in the vaccines. Then the next experiment that we did is we compare these different versions of recombinant proteins in immunized mice. So we immunize mice intramuscularly with these proteins. We include an adjuvant in order to be more immunogenic. This is Adavax, which is an adjuvant that is similar to MF59, which is an approved adjuvant for the use in humans. We use a prime and boost approach in a mouse, and in order to be able to not only look for an induction of antibodies, but look for protection, because the mice are not susceptible to SARS-CoV-2 infection. We took these mice and then we transduce them. We make them to express the receptor, human AC2, in the lungs through the use of a gene therapy vector that is being intranasally given to the mice before challenge. Now, by this gene therapy vector, the mice will express now in their lungs the human AC2, which is the receptor, and then they can be challenged. And although this is not a lethal challenge, one can look to body weight drop for disease as well as to viral titers in the lung of these animals. Okay, so first, if we look to the induction of antibodies by these different vaccines, which are here in the bottom right of this slide. The green vaccine is the wild-type protein, induce antibodies after priming, but the one that contains the red one, the one that contains the cleavage site eliminated, induce higher level of antibodies. The one with only two prolines, the orange-yellow one, induce a little bit less, and the one that has the two modifications induce very good levels of antibodies also. So it looks like the cleavage site elimination is contributed to higher induction of antibodies. These are antibodies that bind to the receptor binding site of the protein measured in an ELISA assay. And we look at post-boost, we have basically the same results. The ones that are giving higher level of antibodies after boosting are also the ones that have the cleavage site removed from the spike protein. So now, if we look now not only to binding antibodies to the spike, here are binding antibodies in the spike in the left, which again are the ones that have the cleavage site removed, the ones that are a little bit higher. But we look to neutralizing activity of these antibodies. Now what we can see is that there is one antigen that is much better in terms of levels of neutralizing antibodies, and it's the one that contains the two modifications, removal of the cleavage site and introduction of the two prolines. And if we now challenge these animals with SARS-CoV-2, then the animals that are not vaccinated lose weight. This is the black line with a peak at 6 days post-infection. And the ones that are vaccinated, in general, they lose also some weight, except with one exception, which is the one that has been vaccinated with the spike that contains the two modifications, both the cleavage site, and the two proteins that make it more stable. This in terms of disease, body weight loss. But if we look to viral titers, then what we see in viral titers is at day 2, we have a reduction in viral titers in all the animals that have been vaccinated, but we don't see in all the animals that have been vaccinated with the spike that contains the two modifications, cleavage site and the two proteins that stabilize it, we don't see any virus at day 2 in the lung. At day 4, they are all equally identical, but it looks that from these studies that the antigen that contains the cleavage site removed of the spike and the two proteins that stabilize further the spike is the antigen that induces better protective immunity in mice. And this leads us now to our studies with the Newcastle disease viral vector. So in these studies, we have also been using a stabilized spike protein to be expressed, not now as a recombinant protein like in the previous experiments, but now expressed by a viral vector. In this case, the viral vector is Newcastle disease virus. So it's similar in terms of using a viral vector to the adenovirus vaccines, but the virus vector that's been used is different. Adenovirus is a DNA virus, Newcastle disease virus is an RNA virus, and it's a virus that it can infect chicken, avian species, especially chicken, where it can induce a very virulent disease, which is called Newcastle disease. And because this was known for a while that Newcastle disease can infect poultry and cause problems in poultry, there is a vaccine that has been developed many years ago against Newcastle disease in chicken, which is based in a Newcastle disease virus strain that does not induce disease in chicken, but administration in chicken will protect them from virulent Newcastle disease virus. So the viral vector that we are using for expressing spike is based on these vaccines of Newcastle disease virus, these vaccine strains that are being used for vaccination of chicken against Newcastle disease. Therefore, they are safe for chicken and are known also to be safe for humans, which is also very important. And most of the studies that have been done with this vector in our team has been done by a very talented person, Wina San. That was the one that was rescuing these viruses and using viral vectors and using them for vaccination in the different animal models. Now, why Newcastle disease virus? What are the potential advantages that this vaccine platform may have? First, there is no pre-existing immunity in humans against these viral vectors. Viral vectors may have a problem that if there is an antibody in humans, in people that have been vaccinated against the viral vector that is being used, then the viral vector will not be able to deliver the genetic material that is required for expression of the spike. This could be a problem, for example, with some of the adenovirus vaccines, for which some people may have some pre-existing immunity against the adenovirus vector that is being used for delivering the spike. Newcastle disease is an avian Newcastle disease virus, an avian pathogen. It does not infect in general humans, so we don't have pre-existing immunity against the viral vector. The most important potential advantage that this technology has is that Newcastle disease virus vaccines, the ones that are used in chicken, as well as the ones that we have developed now expressing a spike protein, they are manufactured using the same technology, which is egg-based, as the influenza virus vaccines. And this, what it allows, is that any production of facility for influenza virus vaccines could be used for producing a Newcastle disease virus vaccine. And it grows very well in eggs. It can be grown in eggs, and it can be used, the egg material, be used for making the vaccine seeds that are required for vaccination, similarly to what is done with influenza virus vaccines. And there is many countries, even developing countries, that have production facilities for influenza vaccines, and they can use these facilities also to now produce a Newcastle disease virus-based vaccine. Then it's important, safety in humans is important, and clinical studies in humans using Newcastle disease virus, especially it has been used for potential treatment of tumors, have demonstrated a very good safety profile, so it seems to be a safe vaccine platform in humans. And in addition, we have been using Newcastle disease virus before as a vaccine vector for chicken. And in chicken, we have a very good use of, for example, a dual vaccine against Newcastle and against influenza, avian influenza, by a Newcastle disease virus vaccine vector that expresses not only Newcastle disease virus proteins, but also the hemagglutinin of influenza, the surface glycoprotein of influenza. In the past, we have also generated Newcastle disease virus vectors expressing antigens from other different infectious agents, like, for example, respiratory syncytial virus, influenza virus that I mentioned already, Ebola virus, SARS-CoV-1, the first SARS, or HIV antigens, and we have been tested in preclinical animal models, and they seem to induce very good responses against the antigens that are being expressed. And now we have the Newcastle disease virus expressing the spikes proteins of SARS-CoV-2 and the clinical development in several countries. So we hope that in the near future, the clinical development will be finished, and this will be a new vaccine that could be used, more easy to produce, more easy to administer also, because it's quite stable than any other vaccines that are available right now. Well, so then how we do that? Newcastle disease virus has a genome that contains all the viral proteins. What we do is we insert it into the genome of the virus, a new transcriptional unit corresponding to the SARS coronavirus spike protein. And we can do it, we have these stabilizing mutations that I mentioned before, that we know that will increase immunogenicity, removal of the cleavage site, as well as introduction of prolines to stabilize it. But in addition, we can also make one more modification, which will be the carboxy terminal region of the protein, the one that anchors the protein into the membrane. We have made a version in Newcastle disease virus that has removal these transmembrane cytoplasmic domain of the protein and substitution, replacement with the transmembrane cytoplasmic domain of one of the proteins of Newcastle disease virus. And what we achieve with that is we have incorporation of the spike protein of SARS-CoV-2 into the envelope of the Newcastle disease variant. So the viruses, the Newcastle disease virus vectors, not only express a spike when infecting a new cell, but also they have the spike decorating the virus surface. This is not the case with the adenovectors, that they have a viral surface for which they cannot insert the spike. Adenovirus vectors will only express the spike in the places that they will infect. In the case of these vectors, the Newcastle disease virus vector, by doing this trick with the transmembrane domain, not only the virus that the cells that are being infected with the virus when there is vaccination will express a spike, but also the spike is incorporated into the virions. The spike is expressed very well. They stabilize the spikes as compared to the non-stabilized spikes that they get cleaved and they get expressed less. And then in terms of production, they can be put in eggs, produced in eggs, and this results in generation of virions containing the spike here in green, decorating the virions. And because of this modality that they have this spike in the surface, we can test two modalities of vaccination. One is we can inactivate the viruses, and then we can inject it intramuscularly, and that will be similar to an inactivated viral vaccine, or we can use it as a live vaccine, because this virus does not induce disease in humans, either intramuscularly or intranasally, which may allow better protection of the upper respiratory tract. And in this case, not only it will induce an immune response because of the antigen that is decorating the virus, but also because the virus will be able to get into some cells and express the spike, and then induce also a good immune response against the spike. So we are exploring both modalities of administration to see which one produces better protection. So the study that I'm showing here is one of the first studies that we did in mice with these Newcastle disease virus vectors, in which we compare one that contains the transmembrane cytoplasmic tail of one of the Newcastle disease virus proteins, the F protein, versus the one that is not anchored into the wild type spike protein, stabilized, and it's not anchored into the virus envelope. And this intramuscular administration live of the Newcastle disease virus in mice, a prime boost combination, and then we use also two different flavors of the Newcastle disease virus, one that contains a mutation that makes it more immunogenic, which is this L295A mutation. And then when we look for induction of antibodies in these mice that have been immunized, what we see is that they all induce good level of ELISA binding antibodies, especially the last version of the vector. And when we look to neutralization, they all induce also good levels of neutralization, but after priming, perhaps the ones that contain this transmembrane and cytoplasmic tail of the F protein of NDB, which are in red, they induce higher level of antibodies than the ones that are in green, that contains the wild type transmembrane cytoplasmic domain. Now, if we challenge the mice, and in this case we challenge the mice with a mouse-adapted SARS coronavirus, a little bit different from the challenge that I showed you before, in which we put with an adenovector, an adenotherapy vector, the human ACE2 receptor. They have now been developed, mouse-adapted SARS-CoV-2. So we can challenge with a SARS-CoV-2 mouse-adapted that we got from Ralph Barrick. And then we look day 4 for lantires. In the control animals, we see high level of viral titers at day 4, but in all the immunized animals, we see control of viral infection and not viral titers in the lung. And if we look for the presence of antigen in the lung of these animals, we see the antigen here as little brownish cells in the lungs of these animals on the right. But we don't see antigen virus-infected cells in the ones that have been vaccinated with the vaccine vector expressing a spike. All right, so this is intramuscular. We have also performed intranasal immunizations using the same vectors, and then what we see is now in these intranasally immunized mice, we see nice levels of antibodies in sera at week 6 and week 10 after boosting. And if we challenge the animals again, we get a very good reduction at day 2 and day 5 of viral replication in the case of the ones that have the improved vector with the spike, with the cytoplasmic tail and the transmembrane domain replace from the ones of the glycoprotein of NDB. We don't see any virus at day 2 or at day 5 in the lungs of these animals. All right, so this is with live viruses, which are live attenuated. Doesn't work also as a inactivated vaccine. For that, we inactivated the virus and then we give it intramuscularly. In this case, we compare just 5, 10 micrograms and 20 micrograms of viral proteins given intramuscularly with lower levels in the presence of an adjuvant. We use 2 adjuvants here. One is Artotap, and the other is Adavax against the MF59 equivalent, which are 2 clinically relevant adjuvants. And for that, we use lower levels of antigen, 5 micrograms, 1 microgram of 0.2 micrograms. And when we look to serum antibodies, we get good level of serum antibodies, but what you can realize that in the presence of an adjuvant, then you get levels that are equivalent to the ones that are in the absence of adjuvant, but with lower amount of viruses. So 1 microgram plus adjuvant induce the same levels of antibodies that 20 micrograms without adjuvant. And again, if we challenge now these mice, what we see is that especially the adjuvanted vaccine with inactivated with Adavax, all the animals that we tested at day 4 with all the doses, 0.2, 1 microgram, or 5 microgram, they don't have viral replication in the lungs. We cannot detect virus titers in the lungs of these animals that have been vaccinated. So this looks very good. And when we were doing these studies, then the group of Jason McLellan found some way to further stabilize the spike by introducing not 2 prolines, but 4 more prolines in 6 prolines is what we call it, in the carboxy terminal domain of the spike that make it even more tight and more stable, these spikes. So when we saw that, then we compare the ones with 2 prolines with the ones with 6 prolines, and that's what we did in the next experiment. So using a ExaPro, we call it, and when we compare it, we see that if we use the ExaPro, it's even getting higher levels of antibodies than the previous version when the animals are immunized intramuscularly. And we look in these animals, and now this is an experiment with hamsters. Hamsters are susceptible to SARS-CoV-2 infection. So in this case, an intramuscularly administered inactivated vaccine in the presence of Adavax as adjuvant, or CpG, or DOTAP. And then after we get good levels of antibodies, and when we challenge with SARS-CoV-2, especially the vaccine that has as adjuvant Adavax and DOTAP, the animals don't lose almost any weight as compared to the other animals. So it looks like this is a good vaccine for both mice and hamsters. Now, this intramuscular vaccination in hamsters does reduce viral titers, and again, the viral titers, especially to the adjuvanted vaccines at day 2 in the lungs of these animals, or at day 5, we cannot detect any viral titers as compared to control animals. And then if we do intranasal administration in hamsters, we also get very good responses. We get very high levels of antibodies in sera, and the animals don't lose any weight when they are being challenged with SARS-CoV-2 virus. So it works both intramuscularly and intranasal. And the important thing is that if we look intranasally, not only in lungs, but also in nasal turbinates, nasal washes, NW, then we get a perfect reduction of viral titers in the lungs at day 2 and at day 5 in vaccinated animals, but also in the respiratory tract, upper respiratory tract. So we get a good reduction of viral titers, and some animals, no viral titers that are detectable at day 2 and certainly not at day 5. Okay, so how about different variants? So now we have been testing these immunized animals, also mice or hamsters immunized with this vector, which is based on the first circulating variants of SARS-CoV-2, what we call the Wuhan strains or Wuhan-like strains. We test for the ability to neutralize some of the variants, like the B.1.1.7 or the alpha variant, and the B.1.351. And what we see is that you get still very good levels of induction of antibodies, neutralizing antibodies, doesn't drop with respect to the alpha variant, B.1.1.7. With respect to the beta, B.1.351, there is some drop. This has happened also with the other vaccines that are in the market, but this drop is not enough to remove neutralizing capability. So these immunized animals are still protected against variants, and then when we challenge these animals with either the beta variant, which is the one that shows more of a drop in neutralizing antibodies, or also with the alpha variant, the gamma variant P1, then we see also a nice reduction of titers in the mice in the lungs of these animals. All right, so that's all the preclinical data that we have, and this has spurred with funding mainly from different governments of clinical trials in different parts of the world using this vaccine approach. One of them is Vietnam. The phase 1 trials started in March 15. They are comparing different doses of an inactivated vaccine in volunteers for safety and immunogenicity, with and without adjuvant. This is an activated approach. Same thing is being, or very similar studies being conducted in Thailand with a slightly different doses, and but also with an activated vaccine. It's studies that started in March 22. We are starting to get the data from these clinical trials. The good thing is that we have not been able to see any severe adverse event, so it seems to be working quite well in terms of safety, and we are now monitoring the immunogenicity that they are inducing in vaccinated people in these phase 1 trials. And in addition, there is another another trial that has been started in Mexico, in which case we use there a live attenuated approach. So we part of the volunteers, they receive the vaccine intranasally, and again we are starting to get data from these ones with the severe adverse events. They don't seem to occur, at least in this group of volunteers, and the immunogenicity we are just pending for getting this data. So what I've shown you is that antigens that are based on a stabilizase protein by removal of the feeding cleavage site and insertion of prolines are potent immunogens, and these are the preferred antigens to be used in vaccine platforms against SARS-CoV-2. That Newcastle disease virus-based vector that expresses the SARS-CoV-2 that is stabilized by removing the cleavage site and putting 6 prolines, induce potent protection from virus infection both in lungs and nose of hamsters after intranasal immunization. And the new variants are still neutralized by sera from immunized mice, but with some drop of titers for the 1.351 variant, but still protection. And 2 phase 1 trials have started in Vietnam, Thailand, in Mexico. There have been started one with a live approach, and we expect soon also trials to start in Brazil and in the USA, but we are still in phase 1. So it may take still some time before we get results of efficacy for these vaccines. But again, if this turns to be a good vaccine against SARS-CoV-2, that will be a vaccine that is more easy to produce, less expensive than the vaccines that are right now, and more affordable, especially for developing countries, which is where the vaccine is still needed. Okay, so thanks a lot, and if there is any questions, I will be happy to answer.
Thank you so much, Dr. Garcia-Sastre. We appreciate your presentation today. We now will move to the live Q&A portion of our webinar. And if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. It looks like we have a question already coming in from our audience. Can you reuse an NDV vaccine vector multiple times in the same individual and still induce good immune responses?
Yeah, okay, so this may have to do with the need for potentially future boosters, as well as potentially using also Newcastle disease virus vectors for vaccination, not only against SARS-CoV-2, but against other emerging pathogens or even some pathogens like RSV. Usually the problem with using viral vectors repeated times against multiple pathogens later on or through boosters is that these vaccine vectors also induce immunity against themselves, and then there is a moment that they are not anymore able to induce good immune responses. Now, this could be a problem for Newcastle disease as use of a general vaccine platform against multiple agents. But the good news is that within the Newcastle disease virus family, there are a lot of antigenically different viruses that are also all infecting avians, and they are safe in humans. So one can change the surface glycoproteins of Newcastle disease virus with the surface glycoproteins of related viruses, which are called avian paramyxoviruses, that will make the viral vector again being able to work efficiently in people that have already antibodies against the previous Newcastle disease virus vector that was vaccinated with. So we hope that with that we can bypass the potential vector immunity that is created when we first use one viral vector, and because there are more than 20 antigenically different avian paramyxoviruses, there is at least 20 different possibilities to make reuse in different vaccination approaches in the same individual, the Newcastle disease virus vector.
Thank you so much for this opportunity to do a Q&A with you. And I want to encourage our members that are attending to submit any additional questions. That any of them that are not answered today will be answered via email. Would you like to provide any closing remarks for our audience before we go?
Well, I think it's important, at this moment, to increase the levels of vaccination. I mean, there is a lot of fear, perhaps, that vaccination is not working well against the new variants, and that there is waning of immunity that may actually make people that are vaccinated vulnerable again. There is a lot of Delta variant circulating now in the world, but perhaps the vaccines are reduce efficacy, although not completely. The vaccine that we have right now reduce efficacy, although not completely lose it, against asymptomatic and mild infections, again infections of the upper respiratory tract, which makes a problem because there is some virus transmission, and especially non-vaccinated individuals, they can cause severe disease, but they're still extremely efficient with respect to elimination of severe disease. Meaning that if we get enough coverage of vaccination, we will reduce dramatically the cases of severe disease. The virus will be still circulating because the vaccine is not 100% efficacious, especially, as I mentioned, against the new variants, but they will eliminate most of the cases of severe disease, making now this virus less harm, producing less harm than what it can produce. So basically right now, the majority of the cases of severe disease are happening only in not vaccinated individuals. So it's like the pandemic now has shifted from being a pandemic of everybody to being a pandemic in non-immunized individuals. And we need to do everything that is possible to protect the people that are not immunized, increase the vaccination coverages in the places that they can afford vaccines, and find new vaccines that may allow to increase vaccine coverages in the countries that they don't have the capability to get vaccinated with the existing vaccines, which is what we try to do with this Newcastle disease virus vector vaccine.
Well, thank you very much, Dr. Garcia-Sastre, for your time again today and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions, again, we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the registration. And this webcast can be viewed on demand for 2 years until September 9th, 2023. LabRoots will alert you via email when it's available for replay. We encourage you to share that email from your colleagues who may have missed today's live event. Bye-bye, everyone.
Okay, goodbye.